Immunotherapy is an innovative treatment that dynamically modulates the immune system to attack cancer cells. Development of immunotherapy is crucial to reduce the usage of other cancer therapies that have adverse side effects. For instance, chemotherapy using alkylating agents can destroy bone marrow cells and increase a patient's risk of getting leukemia. This raises the need for an alternative treatment with less harmful side effects. In this 21st century, chimeric antigen receptor T-cell therapy, or shortly known as CAR T-cell therapy, has electrified the oncology field. CAR T-cell is one of the most promising and advancing adoptive T-cell therapy. CAR T-cells are genetically engineered, stronger and smarter version of natural T-cells. To know what CAR T-cells are, we need to understand their production and structure. To produce CAR T-cells, normal T-cells are purified and transduced with an engineered chimeric antigen receptor gene using either retroviral vectors or CRISPR-Cas9 gene editing technology. This antigen receptor gene is later expressed on CAR T-cells. There are three basic parts to CAR T-cells receptors: an antigen recognition domain, a transmembrane domain, and an internal signal transduction domain. The antigen recognition domain is derived from monoclonal antibodies. Its single-chain variable fragment consists of a light chain and a heavy chain. This confers to the different antigen binding specificity of various CAR T-cells. Next, the internal signaling domain is composed of an activation domain and co-stimulatory domain. The number of co-stimulatory domains varies between different generations of CAR T-cells, conferring to their functionality. Up to the year 2021, there have been 5 generations of CAR T-cells. Each generation has enhanced cancer-killing characteristics. Every generation of CAR T-cells has very similar antigen recognition domain functionality. Firstly, this domain has enhanced avidity to specific tumor antigen. Its antigen recognition domain enables major histocompatibility complex MHC independent antigen recognition and binding of CAR T-cells to kill specific tumors, as opposed to conventional T-cells which are dependent on poorly immunogenic MHCs. This also enables the identification of tumor cells that have evaded immunosurveillance by downregulating MHC class I expression. Secondly, CAR T-cells are designed to recognize a variety of antigen structures such as proteins, carbohydrates, and even glycolipids. However, conventional T-cells can only recognize peptide antigens. Next, the internal signal transduction domain of CAR T-cells, which varies between different generations of CAR T-cells. First generation of CAR T-cells utilized an intracellular domain which was derived from CD3 zeta chain of the T-cell receptor, which induced cytotoxicity against targeted malignant cells but failed to support CAR T-cell expansion in vivo after reinfusion. Second generation of CAR T-cells in the year 2013 had one additional co-stimulatory domain derived from either CD28 or CD137. This co-stimulatory domain recruits various signaling molecules and induces interleukin-2 production. This enabled the CAR T-cells to survive in vivo. The second generation of CAR T-cells were the first 2 CAR T-cell therapies to be approved by the FDA in the year 2017, namely tisagenlecleucel and exicaptogen ciloleucel. Both cells identify CD19 surface antigen on B cells and kill the cancerous B cells. It is used to treat aggressive B-cell leukemia and lymphoma. In July 2020, brexucaptogen autoleucel was approved by FDA to treat advanced mantle cell lymphoma. The third generation in the year 2014 had 2 additional co-stimulatory domains. Thus, it had enhanced cytotoxic activity for tumor cells. Next is the 4th generation of CAR T-cells, also known as TRUC. TRUC is an acronym for T-cell redirected for universal cytokine-mediated killing. These cells are still being researched in trials. TRUCs are based on 2nd generation CAR T-cells added with inducibly expressed chemokines. TRUCs have 4 main advantageous elements to it. Firstly, TRUCs can synthesize cytokines such as interleukin-12. Advanced cytokine-armored CAR T-cells retain their stemness. Cytokine accumulation can transform a cold tumor into a hot tumor, promote memory T cell formation, increase CAR T cell persistence, and even reactivate host T cells to regain its anti-tumor efficiency. Secondly, in fourth-generation CAR T cells, PD-1 inhibitory receptors are blocked by PD-1 blockers, or PD-1 receptors are genetically disrupted to prevent PD-L1 ligand binding, which will cause apoptosis. PD-1 blockers also rescues other T cells to reconstruct the immune system of the patient. Cytokines and PD-1 receptor inhibition enables CAR T cell to resist the tumor microenvironment and prevent its exhaustion, which increases its killing efficiency. Tumor microenvironment is an emerging cancer hallmark that is challenging many cancer treatments nowadays. And this fourth generation of CAR T cells are able to overcome this challenge. Thirdly, artificially transduced CXC chemokine receptor type 2 guides CAR T cell to the tumor that expresses chemokines at high levels. Fourth and finally, bispecific T cell engager BiTE facilitates CAR T cell and host T cell infiltration into tumor sites. Very recently, many studies are being conducted on the next generation, or also known as the fifth generation of CAR T cells. These cells are designed to prevent their terminal differentiation and to have better persistence. Currently, these cells used to design CAR T cells are collected from the patients themselves. It is called an autologous treatment. However, researches are being conducted to use relatively healthier T cells from donors to design the CAR T cells. This is called an allogeneic treatment. Benefits predicted from allogeneic treatment are fast off-the-shelf product availability, less expensive, and more potent treatment. CAR T cell therapy alone in Europe costs about 400,000 USD, which is approximately 1.61 million Malaysian ringgit. However, Malaysian Genomics Resource Centre Berhad is aiming to make CAR T cells available in Southeast Asia for approximately 200,000 Malaysian ringgit only. To conclude, CAR T cell therapy has paved a new path in cancer immunotherapy. Its side effects are more manageable compared to other cancer therapies such as chemotherapy. Therefore, CAR T cell therapy shines brighter hope for cancer patients to fight cancer. Thank you.